hear from Anna Matchinga. She is a senior biochemistry major here at NKU. She is originally from Park Hills, Kentucky, and her plans for after graduation are to attend veterinary school at Auburn. Today she's going to talk to you about beta toxins of the Staphylococcus aureus, which has been known to cause mutations and defects in the heart valve and shell. So she's going to talk to us about that today.
So like Emily said, I will be discussing the impact of beta toxin activities, two main activities, on infective endocarditis. So first I want to give a background just on human infective endocarditis in general. This is mainly an infection of the valves, and these are both prosthetic and native valves, and the lining of the heart, and it's characterized by vegetation. So these vegetations are kind of cauliflower-like structures that are composed of bacteria and other host factors. So here's an example here, a picture of the heart, and then those yellow structures right there are the vegetations. Frequently, pieces of these vegetations will actually break off and travel throughout the body, causing systemic embolization, so blocking of blood flow, as well as metastatic infections, so infections in other places in the body. Overall, this leads to death in 66% of patients that are diagnosed. And Staphylococcus aureus is the most common pathogen that causes this healthcare-related infective endocarditis, as it's responsible for about 40% of diagnosed cases. So beta toxin from Staphylococcus aureus is one of its main virulence factors. It contributes to its colonization and development in the body, and it allows it to survive in the bloodstream. There are two main mechanisms of action that this beta toxin acts by. The first one is the sphingomyelinase activity, and this is its ability to lyse erythrocytes by hydrolyzing sphingomyelin, which is a plasma membrane lipid. The second activity is this DNA biofilm ligase activity, and this is its ability to cross-link itself in the presence of DNA, forming a biofilm. So we know the two mechanisms that beta toxin acts by, but we don't know exactly how they contribute to infective endocarditis specifically. So here are the main goals of the experiment. They first need to better determine the active site of DNA biofilm ligase. They know the sphingomyelinase active site, but they're not entirely sure of the biofilm ligase, so they do a couple tests to better determine the amino acids that are needed for that active site. So to do this, they made several mutants, and they also tested them for retained sphingomyelinase activity to make sure that the active sites were separate and that one activity didn't impact the other. They then went on to determine the contributions of each of the two activities, the biofilm ligase and the sphingomyelinase, and how they contributed to infective endocarditis and sepsis in a rabbit model. So first they made this RN450 delta HLB nuc minus and col HLB minus. So RN450 and col are two common lab strains of Staphylococcus aureus, and this HLB is the gene that contains the beta toxin. So this col HLB minus was previously isolated. They used a bacteriophage to knock out the HLB gene, so the beta toxin activity was no longer in that mutant. They then made this RN450 nuc minus strain, and nuc contains thermonuclease, which is an enzyme that breaks down DNA and RNA. So they used this strain for the DNA precipitation assay so that they would be able to see the oligomerization of DNA without that enzyme breaking it down. So here they look at the expression of beta toxin in those two strains that they made. So the HLB gene from RN4220. So here is like the bacteria, the thing there, and then the red here is the HLB gene. So they amplified that gene by PCR, specifically targeting for it, and then they ligated it into this plasmid PCE104. So then when they added it to the plasmid, they could then transform that plasmid into E. coli. The reason that they added it to E. coli is because there are many, many more ways of manipulating E. coli than there are other bacteria because it's so common. So by putting it in E. coli, they could then use this quick change mutagenesis kit to make their mutants. And after they created the mutant they wanted in E. coli, they put it back into Staphylococcus aureus through electroporation, which is where they submitted these samples to an electric field, which caused the plasma membranes to be less strong and for the plasma to be reintroduced into that bacteria. So here's just a quick overview of that mutagenesis process. So they have the original plasmid here, which is double-stranded like the DNA, and they have two primers. Each of them contain the desired mutant. So one primer anneals to each strand of the DNA, and when they mix it with the DNA polymerase, then here we get the new plasmid with the desired mutants in it. Now, unfortunately, not all of the plasmids take up the desired mutant, so they're still left with some here that are the original ones. So in order to get rid of those, so they only end up with the plasmid that they want that contains that mutant, they digest it with this DPN1, which selects for just methylated DNA, which the original plasmid will have methyl groups hanging off of them. So that DPN1 selects just for those and breaks it down. So then we're just left with our desired mutant plasmid. Once we have that plasmid that we want, then they can transform it, like here, back into Staphylococcus aureus, and then perform the experiments on the mutants. So these are the 6 mutants that they made, and we can go over them further in the coming slides, but they made 6 where they substituted out these amino acids for other ones in the sequence that they knew the ligase activity was most likely contained. So here's the amino acid sequence for B-Toxin. This highlighted portion right here was suggested by a previous paper to be where the DNA biofilm ligase activity is within. So they tested they changed these 5 amino acids with asterisks under them and substituted them for other ones to test to see if those specific amino acids contributed to the ligase activity. Eventually they saw that these 3 that are in white were the ones that were essential to that activity. And then down here, there's a little asterisk next to this H, and that's where they mutated it to make a sphingomyelinase mutant that they used later in the experiment. So now they did a DNA precipitation assay. So they took their wild type in each mutant, and they mixed it with this purified plasmid DNA and ethidium bromide. So they by mixing the wild type of the mutant with the DNA, and then they would centrifuge it down to see if that mutant could oligomerize in the presence of the DNA. So the way they could see this was just visually if a pellet formed in the bottle of the centrifuge tube. So here we can see the results. So the wild type right here that contains the B-Toxin, we can see that little pellet right there, which confirms that that could oligomerize in the presence of DNA. These 2 mutants also had that ability, so they had the pellets as well. But these 3 here did not. So they had no pellet formation, so they knew that those amino acids were necessary for the ligase activity of the B-Toxin. So next they went on to confirm the sphingomyelinase activity. So they made sure that while affecting the ligase activity, they didn't also interfere with the sphingomyelinase activity. So this is a sheep erythrocyte plate, and they streak their samples in them, and that white area around the streak is the bubble of lysis. So that's showing that they could continue to lyse those red blood cells. So these 3 are the 3 mutants that could not oligomerize DNA in the last slide. They didn't have that pellet formation, but they still retained the ability to break down those red blood cells. So they knew that the 2 activities were separate. So next they did this biofilm formation assay, and they grew their cultures in this tryptic soy broth that they enhanced with glucose to give them more nutrients to grow further. So we have our broth here in this side, and then these are the wells of the plate that they previously coated with 20% human plasma so they could mimic the interior of blood cells in our body. So when they added the 200 microliters to the wells of the plate, They let it sit overnight, and then they had to remove 150 microliters and add back in more buffer, because over time, those nutrients get used up. So in order to allow the samples to further grow, they removed some of it and added back in more of the broth, so it had more nutrients and was able to further grow overnight again. They then measured the absorbance of them at 562 nanometers. This is about halfway through the process, just to make sure that they were getting the desired growth of cells in addition to the biofilm that they wanted. So they then removed the cultures from the wells and washed them 3 times with PBS. So after removing the samples, they added 100% ethanol to allow the biofilm to adhere to the sides of the wells. And this is because in our body, the biofilm is in an aqueous environment. So they're recreating that aqueous environment and making it soluble so that it can adhere to the sides of the plates. So after letting them air dry, each biofilm was then stained with crystal violet and rinsed again with the PBS 3 times to remove the excess cells that they didn't want so that they could only see the biofilm formation. And they then treated it with this 80% ethanol, 20% acetone to remove the excess crystal violet so that it wasn't overpowering. They could still see what they wanted to see. They then mixed and diluted with more of this ethanol acetone solution to further get rid of the excess crystal violet. And then they measured the absorbance at 562 nanometers because that's the absorbance at which purple light absorbs. So here are the results. So we can see here this COL and the COL HLB minus plus HLB. So these are both like the wild types. For this one, they knocked out that HLB gene and then they added it back in with the plasmid just to make sure that they were getting the desired beta-toxin activity by adding that plasmid back in. And then this is just the bacteria without beta-toxin. So we can see a difference between the positive here or the negative on each side and the positive result here. So these are the 3 mutants that originally lost that ability to oligomerize DNA. So they went on and further tested them, and we see that 2 of them don't form biofilms like they did in that DNA precipitation assay, but this 3rd one here produced a lot of biofilm. And it's not really uncommon for this to happen because of the way that the experiments are undergone. So originally, before, they were just testing the proteins to see how they acted, and here they actually put them back into an organism, so it was a completely different environment. So it wasn't that unexpected to see a different result. And then this is a graphical representation of those results. So we can see the difference between the 2 wild types here, and then how these were similar to the bacteria without the beta-toxin. So now they did this protein production. So the beta-toxin that they used in that DNA precipitation assay was collected by, again, mixing it with ethanol and letting it sit overnight. So they precipitated out the beta-toxin and then resuspended it in deionized water. And Image J, which is a computer program, was used to measure the diameter of hemolysis. So they then, again, measured on slides that were pre-coded with, again, their sheep erythrocytes, how far each of the mutants could lyse erythrocytes. And they used this to understand further the protein conformation. So they're checking to see if they're changing the structure of the whole protein by changing that one single amino acid. So then they made these 3D models. So we can see the wild type over here and the mutagenesis version over here, and they're very, very similar. The pink region is the sphingomyelinase, the residues that they altered for the sphingomyelinase activity, and this blue portion is where they altered for the DNA biofilm ligase activity. So they realized that by just changing these amino acids, they weren't completely changing the protein structure, and it was remaining fairly intact. So next, they treated each of their samples with trypsin. And trypsin is an enzyme produced by the pancreas that breaks down most everything it comes in contact with. So they mixed each mutant and then the wild type with this trypsin and observed it over time. So they were seeing if the mutant's degradation by trypsin was similar to that of the wild type. So then they mixed each sample with the SDS page buffer and assessed all of the results via SDS page. So a quick overview of SDS page here. We have our sample gel in here, and it's got these little wells at the top. So you can see the pipette coming down and loading the sample into that individual well. And then a current is sent through from the negative electrode to the positive electrode, and the mixture of molecules is then separated because of that current. So they've got this porous gel, and at first all of the molecules are sitting at the top. Once it undergoes electrophoresis, they then are separated out so that the smaller molecules travel further because they can easily fit through the pores in the gel, whereas the larger molecules stay more at the top. So they're able to see different bands of molecules on that gel so they can distinguish between how these are breaking down in this case. So here are the results. So on the left side here we have the wild type and then 2 of the mutants without trypsin. So they tested this H162A, which was one of the mutants that oligomerized in that DNA precipitation assay, but also didn't have biofilm formation in the biofilm assay. And they also tested this D163A that originally could not oligomerize DNA, but then it did have that great biofilm formation in that second assay. So we can see here there's one dark line all the way across of the protein. So, and it's pretty similar in all of them. There's not a lot of other bands surrounded by it. And then when they mixed it with trypsin, we can see it's breaking down. So this band is not nearly as dark or defined. And there's a lot of smaller little bands underneath it showing that that's being broken down into smaller pieces. And they said that the rate at which these break down was very similar. The 2 mutants were very similar to the wild type. So they concluded that their stability was similar because of this, and they weren't completely destroying the protein by changing those amino acids. So far, they looked at mainly these 3 mutants, and they didn't, all 3 of them did not have the biofilm ligase activity, and all 3 retained that sphingomyelinase activity. And then the biofilm formation was where it got different. So the 2, the top and the bottom ones, both had ones that were less than the wild type, and that middle one was the one that still formed that biofilm when it was put back into the organism. So then they characterized in this last assay the protein stability. So they didn't test this last mutant because it was so similar to the first one, but they concluded that both mutants had similar protein stability to the wild type. So this helped them to better characterize the DNA biofilm ligase active site. They knew some of the amino acids that were important for that activity. So now they wanted to determine the contribution of each, the sphingomyelinase and biofilm ligase activity, and how it affected infective endocarditis and sepsis. So they did this in a rabbit model. They anesthetized the rabbits and inserted the catheter into the left carotid artery until the aortic valve was reached. And they left it there for about 2 hours to induce damage. And the reason they did this on the left side instead of the right side is because in our hearts, when the veins bring blood to the heart, they go into the right side, and that blood is poorly oxygenated. So after it's brought into the heart, initially it's taken to the lungs, oxygenated, and brought back to the left side of the heart. So now the blood in that side is oxygenated, and Staphylococcus aureus is a facultative anaerobe. So it can exist in both aerobic and anaerobic conditions, so both with or without oxygen. But it's able to exist and act a lot better when in the presence of oxygen. So they did this on the left side so that it would better be able to act and they could analyze the results more accurately. So then they closed that incision site and injected the bacteria through a vein in the ear. So this is just a little visual. You can see the artery in the middle, and they injected it in this marginal vein along the ear, and then it was taken to the heart and then the site of where the damage was created. So the rabbits were then treated with erythromycin, and most plasmids have a antibiotic resistance gene. So the plasmid that they used was erythromycin resistant. So the reason they treated with erythromycin was to then select for just that plasmid, so that they knew that that was the only thing acting and contributing to the infective endocarditis. The rabbits then, the experiment lasted 4 days. Some of them died before the end of the experiment from sepsis, but if they hadn't died at the 4-day mark, then that was when they were euthanized. And they removed the hearts and analyzed the vegetations that they saw formed in the bacteria in all of those vegetations. So they plated the bacteria and determined the colony-forming units and the total number of vegetations. So the first experiment they did, they used this lethal dose to a 50% endpoint. And you can see here the wild type, all of the rabbits were dead within 1 day. And this is the organism without the beta-toxin. So it was still lethal, but not nearly as much as the Staphylococcus aureus with the beta-toxin activity. We can see here in this top mutant, this 289N was a sphingomyelinase mutant. So when they knocked out that sphingomyelinase activity, the lethality was completely lost. So that was really interesting because even here with the DNA biofilm ligase activity, it was still fairly lethal even without that activity, but without the sphingomyelinase, the lethality was gone. So next they did an experiment where they did a lower inoculum, so it didn't kill the rabbits, and they could further analyze the size and the bacteria and the vegetations that were formed. So here we can see the wild type. Here is the vegetations. There are a lot of vegetations, whereas here is the Staphylococcus aureus without the beta-toxin, and the level of vegetations in that was really similar to all the mutants. So this one on the right was the sphingomyelinase mutant, and these three were the biofilm ligase mutants. So all of them were deficient in their ability to produce vegetations, and since they were so similar to this, the bacteria without the beta-toxin, their activities were not additive in forming the vegetation. So each of them were responsible for the vegetation formation. However, even though the number of vegetations was so different, the colony-forming units total there was no statistically significant difference in the number of colony-forming units that were counted from each of the samples. So what this suggests is that while both activities contribute to vegetation or the vegetation growth, by increasing the growth, they're recruiting more host factors and bacteria to adhere there and cause more damage rather than specifically causing the damage themselves. They're increasing the surface area and giving it more area to cause damage. So overall, they determined that the sphingomyelinase activity increases vegetation size, but they're still not exactly sure how. They're not sure what it is that's increasing that. They think it could potentially be that since they're breaking down that sphingomyelin into those parts that it breaks it into, that's just creating surface area, and that's what's giving the platform there for it to build on. And they also saw that the sphingomyelinase mutants yielded decreased vegetation size, even with the DNA biofilm ligase activity intact. So this shows that maybe without the sphingomyelinase activity, the biofilm ligase activity doesn't have enough of a platform to build upon. So in the future, they need to specifically characterize what it is about that sphingomyelinase that is inducing the inflammation. And they also need to determine what exactly the host factors are that the DNA biofilm ligase active site binds to. Once they do those two things, they can eventually apply this work to humans and maybe create therapeutic agents to target these things. I would like to express my appreciation to Dr. Guy, my mentor, and Dr. Hare, as well as the whole NKU chemistry department. That they did, so a fair amount, I'm sure. Yeah.
So this may be a dumb question, they covered it. Did they choose a specific type? Because I know like there's specific types of like mice and that kind of stuff. Is there like, was there a specific type of rabbit or especially for that rabbit that they chose?
I think rabbits in general they chose because their system, at least their heart, mimics the human heart pretty well. So then they were able to better characterize their results and apply it more to human medicine to further come up with like these therapeutic agents to help treat this. So that's why they chose that. Yeah.
So like with the heart like constantly moving and pushing fluid through it, like how is the bacteria like able to form a vegetation on it? Why would it not just be pushed through and kind of clump somewhere else?
So a lot of times with these infections, they come from some type of injury. So in this experiment, when they put that catheter in and left it there to create the damage, when they closed the rabbit back up, the immune system would then send like their response to try to fix the damage. And that includes fibrin, which is a protein, that long fibrous protein that kind of makes a mesh to help clot the blood. And Staphylococcus aureus itself has a specific binding affinity for that fibrin. So that's why Staphylococcus aureus is so common in this, because it binding to that fibrin allows it to remain there and form the vegetation.
Faculty. Okay, so I have one. So they went through a lot of the mutant ones with the rabbit studies. Did they do any, or did they mention anything about basically staph infections without the heart damage? Did they do any controls like that, or heart damage without the staph?
They didn't, but I read that there are several different levels of severity of these infective endocarditis. So it can result from that specific injury in the heart like they did in this experiment, but a lot of times, since it can be so metastatic and go from anywhere in the body, they said there are some low-level ones that could even come from damage in your gums or something like that, where it could eventually snowball into this huge infection and sepsis.
Nothing else? All right, well, let's thank our speaker. And we'll be back on Monday for